Our research is on the development of nanoparticles for the targeted delivery of erythropoietin receptor (EpoR) plasmid to the ischemic muscles. EpoR was selected because it facilitates stem cell recruitment, cell protection, and angiogenesis. It will be delivered via anti-intercellular adhesion molecule 1, anti-ICAM1, conjugated to PLGA nanoparticles, as ischemic areas highly express ICAM1. Peripheral arterial disease (PAD) is the buildup of plaque in the artery causing reduced blood flow. It is commonly found in elderly and diabetic patients and is one of the leading causes for morbidity and mortality. The current treatments for PAD shows poor therapeutic effects and produce several limitations. Therefore, we hope that our new drug delivery system is able to overcome these limitations. For our experimental setup, first we will culture bacteria with the EpoR gene. Then we will purify the plasmid and fabricate the nanoparticles to encapsulate the EpoR plasmid. After that, anti-ICAM1 will be conjugated to the PLGA nanoparticles using NH2 and NHS chemical conjugation. For our test plan, we will first perform characterization studies of the nanoparticles, such as its size, polydispersity, zeta potential, morphology, and conjugation of anti-ICAM1. Then we will perform in vitro studies on activated endothelial cells to observe the nanoparticles' cellular uptake, flow study, transfection, angiogenesis, and cytotoxicity. For the in vivo study, the nanoparticles will be injected into mice with PAD intravenously to assess the biodistribution and therapeutic effect. Our result in Figure 4 shows that the purified EpoR plasmid successfully transfected the cells, where GFP, a gene sequence in our EpoR plasmid, was expressed by lenti-X cells. Figure 5 shows the flow cytometry test in endothelial progenitor cells (EPC), where the EpoR loaded PLGA nanoparticles have an increased GFP intensity than blank PLGA nanoparticles and cells with no treatment. Figure 6 confirmed the upregulation of ICAM1 on EPC when stimulated by tumor necrosis factor alpha, TNF-alpha. And lastly, for Figure 7, the API is the stain for nucleus and now red for PLGA nanoparticles. The first column is blank PLGA nanoparticles with TNF-alpha activated cells. The second column is anti-ICAM1 conjugated PLGA nanoparticles with TNF-alpha activated cells. And the third column is anti-ICAM1 conjugated PLGA nanoparticles with unactivated cells. As can be seen, there is an increase in uptake of anti-ICAM1 conjugated PLGA nanoparticles with TNF-alpha activated cells. To conclude, our in vitro results show that our drug delivery system is able to transfect and actively target activated endothelial cells. Additional in vitro studies such as angiogenesis and cytotoxicity, and in vivo studies such as biodistribution and therapeutic assessment should be done in the future. Our current progress in the study is to assess the capability of this drug delivery system at targeting ischemic sites in mice animal models by loading ICG.